introduction, and then the stage will be yours, Summit. Okay. Hi again, everyone. Thanks for everyone for joining us tonight, and especially thanks to Thomas for accepting our invitation to be our next speaker in this webinar series. So I'll just do a brief introduction to our speaker tonight. Dr. Thomas Parr is a theoretical neurobiologist and practicing physician. He completed his undergraduate medical studies and PhD at the University College London, where he worked with Professor Carl Frisson at the Wellcome Center for Human Neuroimaging in Queen's Square. He is interested in how our brains model our environments and how these models become dysfunctional in neurobiological diseases. He's an author of the first comprehensive textbook on active inference, which is an approach to understanding brain and behavior from first principles. He currently works as an NIHR academic clinical fellow in neurology at the Nuffield Department of Clinical Neuroscience, University of Oxford. Once again, thanks for accepting our invitation, Thomas, and the stage is yours.
Well, thank you very much for both the invitation and for the introduction. So I was trying to work out what to talk on today, and I thought it'd be interesting to focus on an anatomical perspective on things, and I'll sort of try and justify why that's relevant from an AI perspective and sort of the links between anatomy of a generative model of the sort that we're seeing used more and more in the context of artificial intelligence, and the way in which we understand the brain in terms of generative models and what that says about the structure and function of the brain. So the sort of broad approach I'm going to take in this talk is I'm going to start by describing why we might think of neural dynamics as a process of inference about the outside world. And I'll sort of talk you through a very simple example of a physical system that sort of makes some of the ideas there a little bit clearer. From there, once we say that the brain is solving an inference problem, we need to specify what that inference problem is, and that's where the generative models come into play. Now, generative models imply both a particular means of solving an inference problem in terms of sparse distributed message passing that takes us directly through to concepts of anatomy, of both sort of microcircuitry anatomy, but also the connectivity between different brain regions. They also, under certain assumptions, imply particular patterns of behavior. And I think that, again, is sort of really interesting, that the same underlying model makes predictions in two different domains, both in the context of the structure you might expect in the brain and also in the behavior you might expect it to exhibit. And so both of these things are things we can measure to get at the models that our brain is using, and potentially use those to influence the sort of models we might design in artificial intelligence. And then finally, one of the things I find very interesting is the way in which we can use pathology and understand how brains go wrong as a way of informing each of these things. Because if you imagine that a brain is solving a particular kind of generative model, and then you disrupt that brain with a stroke or a neurological lesion of some description or a psychiatric pathology, then that will influence the kind of generative model you might see. And we can measure the influence of that, both in terms of the anatomy of where that lesion is, and in terms of the behavior that results from that lesion. So it gives us a way of measuring the effect of lesions on a generative model, and therefore a way of pinning down both the mechanisms of the generative model and how it might relate to anatomical structures more directly. So let's start with this idea of neuronal dynamics as performing a kind of inference. And I think this is something that's often quite tricky to get your head around until you simplify it to a really simple kind of physical system. And so the physical system I'm showing you here is a system of 3 beads connected to one another by some springs. So we can imagine that the blue bead in the middle is connected to a central point by the blue spring. The green bead is connected to the blue bead by the green spring. The red bead is connected to the blue bead by the red spring. So as they move further apart, they get pulled closer together. As they move closer together, they get pushed apart as the spring gets compressed. So this is just sort of very sort of basic Hooke's law type Newtonian dynamics. And what I'm showing at the lower part here is a three-dimensional plot showing the locations of each of these beads over time. And one of the interesting things you see here is that the trajectory seems to always move around this red line in the middle, that there is a sort of consistent distribution over time that you might expect to see these beads in. So let's say we start by initializing this system from a whole series of different random initializations, and then look at the distribution we might get if we plot their trajectories. You can see there's a very clear distribution around that red line in the middle. That's really important because that tells us quite a lot about the relationship between each of these different parts of the system from one another. And if we look specifically at the red and the green beads here, we can say that given the position of the green bead, we can now predict some distribution over the position of the red bead. In other words, we can start to interpret the dynamics of the green bead, which doesn't see or have any direct access to what's happening with the red bead, as performing some sort of inference, as encoding some sort of beliefs about something that's happening for the red bead. And if we plot those beliefs there, we can see they may or may not be correct all the time, but on overall, they capture the positions that the red bead will be in most of the time. So we've sort of reformulated a description of the dynamics of the green in terms of beliefs about the red. And we can make an analogy there between different states of the brain, the things that the brain uses to interact with the outside world and the outside world itself. So this brings in this idea of a Markov blanket or boundary by which our sensory input and our motor output, represented here in blue, mediate all interactions between inside and out. But because we've looked at those relationships between what's happening on the inside and out via these blanket or blue states, we can interpret the internal states of the brain as representing some beliefs about something that's happening in the outside world. So here we have that. I'm sorry, I'm just going to change where I'm going to just reduce the what I can see from the video so I can see this clearly. So yes, no, we're now saying that something on the inside internal states mu encode beliefs about the external states here eta. Now, we can interpret this further as saying that this is a bit like the green states representing a Bayesian belief about what's happening in the outside world given some model. So we're saying that our beliefs about where the red bead might be, this eta given mu, will approximate under some generative model the beliefs that I would have given observations about the blue bead about where the red bead might be. So beliefs about the outside world given my sensory data, for instance. And that model will be made up of prior beliefs, which are shown with this top black square, influencing beliefs about where the external states of the world might be and how those would predict, so this lower square, the blanket states or sensory data that I might expect in the lower part of this model. And so that's the argument sort of taking us from simple dynamics that may be, or even more complex biophysical dynamics, through to an interpretation of that system in terms of beliefs about what's going on outside of it, and therefore in terms of the solution to a particular kind of generative model. So that's taken us through neuronal dynamics and inference through to this idea of the generative model. And the next question really is what sort of generative models do we have in mind? How do we deal with generative models? How do we represent them? And I'll just sort of give you a brief overview of that. So generative models we will typically represent as being a joint probability distribution over often some data y, and then the series of states or variables that we can't directly observe, but that cause those data. So here x1, x2, x3, etc. Now, it's often difficult to interpret a generative model seeing it written down mathematically, but one of the key things that makes them useful is they will typically factorize into simpler parts. So I talked earlier about priors and likelihoods, mapping from the hidden states or the external world through to some sensory data. But there may be further factorizations that you can achieve with a model of that sort. So here I'm just showing a completely arbitrary model that factorizes into a series of different conditional distributions. Again, it's often difficult to really appreciate this in equation form, but there are useful graphical notations we can use that simplify that. And effectively, the logic of doing that is we take each factor of the probability distribution and we represent that as a square. And then we connect up the different variables that are on the right-hand side, so the variables that are conditioned upon, to the variables that are being predicted by that distribution. So effectively here saying that x1 and x2 are predictive of variable y. We then take the next distribution or the next factor and we put another square in that. And again, you can see the same logic. x3 is predictive of x1. And similarly, x2 is predictive of x4 predictive of x2. And you can go on like this, constructing a generative model of this sort. And you can see it's much easier to interpret what's happening with this model by looking at this graphical interpretation than looking at each individual factor of the probability distribution in equation form. And so we can find a much more useful way of getting that idea across. And this is a sort of broad idea that's relevant for all sorts of processes we use without necessarily even thinking about the generative model. So for instance, a principal components analysis is effectively taking some normally distributed set of data points, stretching them in various different ways by some matrix A to generate the data we observe. And the process of doing principal components analysis is trying to go backwards, so infer the x from the y, effectively working out in which ways we've had to stretch it, the matrix A in this system. Similarly, canonical variance analysis is a very similar kind of process where you start from, again, some cloud of data points that you then stretch in two different ways, either by a matrix A or a matrix B, and you get two different patterns. And the problem is finding the A and B that take you back to the X, thereby working out the directions that or sort of the common data points that have been stretched to achieve the data that you've observed. It's not all in continuous space, so we can imagine having clustering-based models where we have a categorical distribution over alternative states I might be in while generating the data point that then lead to various data points in continuous space. And again, the problem of clustering is going from these data points back to these cluster identities of which of these 5 clusters I might have belonged to. And this also applies to the sort of generative models, although this is a slightly atypical way of representing it, that are used in modern day generative AI. So transformer architecture, for instance, is based around the idea of having a series of priors that are conditional, but each state in a sequence is conditional upon multiple states that have happened before. So you get this very non-Markovian pattern of prediction of the next state. And the states we're predicting are often categorical states like words that rely upon some embedding structure to reach these latent variables. So it may be looking at a vector in a high-dimensional space that's been progressively transformed from x1 to x2, from x1 and x2 to x3, from x1, 2, and 3 to x4, etc. That's then used to predict some token or word in a sequence. And the point I'm making here really is just that this sort of representation is very flexible and deals with a lot of the generative models that we work with, either implicitly or explicitly. And so that's just a sort of brief overview of the idea of a generative model and the ways in which you can use them to represent a whole series of different kinds of process. The next question really is how that then relates to neurobiology and anatomy, that gives us a way of sort of measuring the influence of those generative models in the brain. And the key concept that links them together is the notion of a Markov blanket. And here, this is not sort of using the concept in the same way I did earlier on when I was talking about a separation between the brain and the outside world, although the underlying mathematical formulation is identical. But here it's looking at ways in which we can understand the sparsity of these generative models, the conditional dependencies and independencies. And the idea is that a Markov blanket is the set of states that provide a separation between one variable in a model and all other variables that are in that model. So that will typically be expressed in terms of if we were looking at the Markov blanket for x2, we would look at the parents of x2, the things that are predictive of it, the children of x2, the things it generates, and the parents of the children of x2. So our Markov blanket for x2 is going to include x1, x4, and y. And the key point there is that if we knew those things, then there would be no additional value to knowing about things like x3, that we have all the information we need to predict what's going on with x2 or xi, as I put it here. And that's true no matter which x we choose in our model. We only need to know about its Markov blanket to be able to predict what's going on for that particular variable. And this is really useful in that it helps us derive all sorts of message passing schemes based upon sparse information available from different parts of the model that let us infer what's going on in particular parts. And belief propagation is one example of a scheme that that exploits that kind of Markov blanket structure. So here we're effectively saying that to know about x2, we just need to know about the messages shown by these arrows that are passed across the neighboring factors to x2 itself, and that's just expressed mathematically in the expression below. And that gives us a very powerful way of understanding the inferences we can draw in a generative model, but also has implications for thinking about the very sparse message passing that we see across synapses and axons in the brain. So how do we get from these messages to the kinds of anatomy we might look at in the brain? Well, one way we might approach this, and there are a number of different ways we could do, but one way we might approach this is to say, let's imagine each of these arrows represents a neural population. So we could take the series of neurons that are dealing with the interactions with the data Y. We can then map those to the interactions they have with other neural populations, and similarly for the other variables. So by taking each of those messages, we can develop the same sort of sparse message passing across different populations that we might see across neural populations in the brain. And we can look at that in more anatomical detail by thinking about the kinds of generative models the brain might use. So for instance, let's say that we have some states of the world S that generate some sensory data O. Those states of the world might be generated by slowly changing states of the world and have a sort of hierarchical structure to them. So we contextualize things in this kind of hierarchical structure. At each level of this hierarchy, we might expect to generate dynamics. And at lower levels of our hierarchy, from the superscript 1, we might move from a state at time tau to time tau plus 1. Over a slightly slower timescale at a higher contextual level, we might move again across this sequence, but much more slowly. So, for instance, if the superscript 1 states are different words in a sentence or different letters in a word, then the states at time 2 might be the words or the sentence that are progressing much more slowly, but providing a contextualization that allows us to make those predictions. Then the transitions at each level themselves are going to depend upon the choices we make. And so I'll just put those in boxes to suggest that there are alternative transitions that we might make. We can then start to put together messages and look at how those might relate to the neural populations we see in different cortical layers, which I'm showing on the right. And so the neocortex in many animals, and certainly in primates and humans, is divided into 6 distinct layers. So sort of going from layer 1 that's close to the scalp, and layer 6 that is sort of closer to the deeper parts of the brain. And there's a very stereotyped pattern of message passing among different neural populations there. So let's imagine that our predictions of our observations or data O, given our states S, are represented by these neurons in layer 6, which is consistent with the descending message passing we see from higher order cortical areas through to more sensory areas or areas of the thalamus and other bits of the brain that deal with sensory data. Then we can link together the different hierarchical layers by looking at these sort of more superficial sort of layer 2 type neurons and layer 1 neurons that receive this descending input between different cortical layers. We can then look at the message passing that goes in the opposite direction. So we know that sensory inputs will typically target layer 4 neurons, and sensory thalamus will typically target those layers, projections to sort of layers 3 and 4. And that will pass further ascending messages that go up cortical hierarchies by projecting from more superficial layers to the layer 4 of the next cortical area in the sequence. We then get to this problem of predicting what happens next and dealing with our dynamics. And as I said, these boxes are designed to illustrate that there might be alternative sequences that might come to play depending upon which choices we make. So here we can imagine that we've got several alternative options that might come about. And message passing that comes back based upon the plausibility of those options as well. And so the point I'm making here is that we can start to construct this sequence of neural populations and the asymmetric stereotyped message passing we might see in each area of the brain between that and other areas by understanding the sparsity of this message passing in terms of a fairly generic kind of model that deals with dynamics, so the multiple different timescales, deals with hierarchical inference and prediction of sensory data from those. So that's just sort of very briefly, and we'll come back to a few more details of that in a little bit. But the key thing we now want to do is to say, well, how does the same generative model that might predict synaptic message passing predict behavior so that we have another kind of measurement that we can pin this to? And I'll come back to this idea that I was describing earlier, that we can understand these sorts of systems that are coupled together by some interacting blanket states, sensory data, motor outputs, and understand them in terms of these belief dynamics within the generative model. But while I've talked about the idea that we can understand the green states as doing a kind of inference, it's also important to see them as influencing the blue states here in our spring system by either stretching to bring the blanket states closer to it or compressing the green spring to push them further away. So there's an influence of these belief encoding states on our behavior itself. And one way we can look at that is by thinking about how we might make predictions. So here, let's say the expected value of this likelihood distribution, so where I expect the blanket to be, given my beliefs about the external state. And so here I've just taken the green states out of the system entirely. But remember, both the red distribution here and the blue distribution it implies are implied entirely by the dynamics of the internal states. So they're given the internal states, where do we expect the external states to be, and where do we expect the blanket states to be? I've also taken out the external states here, so that now we're dealing with the system from the perspective entirely from the brain or from the internal part of the system, where it doesn't know where the external states are, but it draws an inference about them. It doesn't know where the blanket states are likely to be next, but it can observe their current value. And we can now start to see this system as one in which the whole purpose is to bring the blanket states in line with predictions. And we can see that when the blanket states don't match this blue distribution, one of two things happen. One is that we adjust where the red states are, or our beliefs about them, so that the blue states fit. So we're bringing our predictions in line with what we're observing by adjusting our beliefs about the external world. But another interpretation of this is that we're actually moving the blanket states, we're moving the blue states in such a way that they match our predictions. So you have this bidirectional changing the world to match my beliefs and changing my beliefs to match the world, that really captures the idea that both behavior and perceptual inference are just two sides of the same coin, the same process, the same optimization of model fit between the internal and external world. And this is really the core idea that is underneath active inference. The point being that the generative model we have in play is now not just a model that we use for inference, but also implicitly guides the behavioral choices we make. And we can interpret that in terms of sort of classical reflex arcs in neurobiology, the idea that effectively our brains are making a prediction that's being projected down into our spinal cord. And that might be, say, proprioceptive joint position data, and taking action that then ensures that our predictions and our data match. And so what I'm showing just in the lower left here is this idea in the context of what might happen if you were to tap on the tendon of the biceps muscle, something that's done in clinical practice to assess someone's reflexes. And the idea that effectively what's happening there is that you're introducing an unexpected change to proprioceptive data that then generates an action to try and bring that back in line with predictions. And that's an explanation for this sort of reflexive action. But in principle, you could provide a whole series of different kinds of prediction depending upon the motor action you want to take, sort of saying that actually a motor command is nothing more than a prediction about the joint position sense I'm going to get from my muscles. So how does that then relate to this idea of these kinds of generative models and message passing? Well, so we're now saying that the descending messages at the lower level are effectively those that are making these predictions that are then enacted by our muscles and spinal reflexes and brainstem reflexes. So working out what we're going to predict becomes really important in terms of deciding what we're going to do next. And for that, we can add in these additional set of states representing our policies or the choices we might make. And this is really interesting because we're now trying to make predictions and inferences about what's happening for things in the future before we've actually obtained the sensory data for that. So let's just add into our graphic here the additional messages we now have to deal with, and the predictions we're now making about future states and future outcomes. So we're now dealing with a situation where we're passing messages into these sort of red boxes, these boxes of future things. And we're effectively treating that as a series of alternative models about what I might do. And the information that's available there is effectively the information that's being passed by these red and blue arrows representing message passing forwards and backwards through time. And we can use those messages as a way of comparing between alternative models of what I might do. And so that's why this sort of red arrow might depend upon those. That's going to be contextualized by our predictions from higher levels. And so we get these additional projections that sort of give two different pathways through to our decisions about what we might do. And messages backwards in time there as well. And these predictions then contextualize the predictions I'm going to make that then generate my muscle actions. And we might see this sort of additional element that we're seeing in the model as representing things like subcortical basal ganglia loops, which are sort of deep structures in the brain involved in the decisions we might make between alternative courses of action. One of the interesting things here is that these sort of two separate pathways, one that's very highly contextualized and deals with all the predictions about what's going to happen next, and this other pathway shown on the right that's much more dependent upon a higher level context, matches quite interestingly to the fact that the basal ganglia receive two different pathways known as direct and indirect pathways through from bits of the cortex that have similar sort of facilitatory and inhibitory type properties, and I'll come back to that in a little bit. But really, the argument we've got so far here is that we can interpret neurodynamics as inference. That implies that if we can write down the right sort of generative model, we have implications both for the behavior we might expect to observe through the predictions of that model through reflexive fulfillment of those predictions, but also about the anatomy that might support that. And that really gives us all we need to start thinking about this notion of pathology and the way in which disease processes might influence both of these things. So there are a lot of different things we could look at here and a lot of different examples where this has been studied. And I'll sort of give you a brief overview of some of the ideas there and then hone in on a couple of specific examples. So one is looking at the behavior we might expect in visual sampling disorders and the intrinsic motivation and priors about how we might act that then might explain things like visual neglect syndromes. And I'll come back to that in a bit. We look at things like the way we model sequences in time and the segmentation from one moment to the next that's really important in this kind of predictive planning process, and the ways in which that might break down in things like Parkinson's disease. And I won't talk about this in any more detail, but the idea is that if we're trying to infer, or our model includes an internal clock that sequences when things might happen, then break down in terms of how we draw inferences about the internal clock states has a really big influence in terms of the kinds of movement we might make. We might look at sort of disorders of memory and the idea of episodic compression. So the idea of sort of patterning things with this separation of hierarchical levels that allows us to deal with sort of episodes that are slowly evolving at a higher level and predict sequences of things that are happening much faster at lower levels of a model, and how that might have implications for disorders like Alzheimer's disease, where the ability to encode different kinds of episodes and then to explain linguistically what's happened during those episodes is impaired. We might think about disorders of sort of eye movement control and coordination that we might pick up clinically by assessing how someone might follow a moving target with their eyes and the implications of that for movement coordination. And that's particularly interesting in thinking about the kinds of models we might use to predict things that or to deal with things like eye movements where you don't move each eye independently. And the idea that maybe there's a single model that determines where I might want to look that has implications for the behavior of both eyes. We might look at the different prior beliefs we might have in terms of how we behave and the kind of executive dysfunction disorders that we see when those priors conflict. And so the example here is a simulation from a Stroop task, which is a classic neuropsychological task in which people are asked to name the colors of the font in which a word is written. But when the words themselves are color words that conflict with that font color, that can cause people to make a series of errors that are interesting to measure and see how the implications that has for the kinds of model they might use. We can look at more communicative type priors, of how we sequence phonemes and words, different kinds of lengths, and the way that might have implications for speech disfluency disorders like aphasias and stuttering. We can look in more detail about the complex problems in motor control and the way in which movement disorders can inform that. And I'll come back to this one as well in a bit. And we can look at things like working memory disorders and the way in which we sequence things over both discrete time to be able to retain a memory of which target I was supposed to look at over a sequence of steps, while also being able to enact a behavioral response to that through a continuous time trajectory. I'd love to talk you through all these things in greater detail, but given time constraints, I'm just going to focus on two of these. So we'll start with this idea of intrinsic motivation and visual neglect. And one of the key ideas of intrinsic motivation is that while there has been a lot of focus over the last few decades on motivation by reward, by the idea that I want to get a particular kind of data, there's, I think, increasing emphasis on the idea of intrinsic motivation, of there being things that are reward-free behavior. And one of the most interesting versions of that, I think, is looking at curiosity and the idea of information seeking. And to give you a bit more detail on that, imagine we're dealing with a model where we're not just modeling how states cause observations, but we're also modeling the relationship between those things. So we're saying, what are the parameters of the distribution that map states to observations in the world? And then that gives us a really interesting thing to try and learn. To make it a little bit more precise, I've just included a couple of equations here, which effectively say that the distribution over these likelihood parameters, these predictions of, given this state, what observation would I see, are represented as a Dirichlet distribution, which is the conjugate prior for a categorical distribution that maps those states to the observations themselves. And to give you a bit more intuition for how that works, well, first of all, I'll just put the equation here that sort of gives us our posterior distribution that allows us to update that. But to make that more intuitive, imagine we're in a situation where we know we're in a particular state. There are lots of observations that we might make in those states. So each bar represents a different observation I could make. We're seeing the observations in this upper part here. Sorry, I don't know if you can actually see my mouse, but the sort of dot that's moving from side to side above each of these graphs. And the idea is that as we make a series of observations, we sort of add to the distribution of how likely we are to see that observation. So each time we see something in a particular state, we increment our distribution for that state, for that observation, and effectively just count up the number of times we've made that observation. And then to ensure it's consistent with a true probability distribution, we renormalize that. So we divide by the total number of observations we've made. Now, there's a really important distinction between the two different graphs I'm showing, because exactly the same data is being shown, but you can see that the distributions are evolving in very different ways. And the difference between them is an assumption about how many observations I might have made in the past, or how confident I am in the distribution at the very start. So we can imagine the upper plot is one in which I've made very few observations in the past, and so every new observation is very, very informative. Whereas the lower plot is almost as if I'm very confident at baseline that I'm dealing with a uniform distribution. And so I get much smaller changes from each new observation. One of the examples I think is often useful to think about this is if you imagine I give you a series of coin flips, and I flip the coin 5 times in a row, and I get heads every time I flip the coin. You might be quite likely to update your beliefs and say this is likely to be a weighted, unfair coin that's always going to give me heads. However, if I now told you that I'd flipped the coin 100 times before that, and 50 times got heads and 50 times got tails, yes, this additional 5 heads might update your beliefs a little bit, but actually much, much less, and you're much less likely to conclude that it's an unfair coin. And really that's the distinction between what's happening in the upper and lower plots here. Now, why does that matter for the idea of sort of curiosity, information seeking, information gain? Well, it's because those states that have a distribution much more consistent with this upper plot are those where I'm more likely to change my beliefs. There's more information to be gained by sampling states of this sort compared to those in the lower plot, where I'm less likely to cause a big update to my beliefs. And I'll show you that in a slightly more specific example. So imagine here we're dealing with a situation where I can sample different parts of a really simple visual scene made of a series of different dots. If I fixate one of the dots, it will change color from black to red. The upper left plot is showing the sequence of samples I might make. So if you imagine this is a simulated eye tracking trace, and the lower plot is showing a simplified representation of the visual data in this very simple kind of visual system. So it's got a sort of inner box that is saying, am I seeing a dot within this box right now? And then slightly lower resolution visual fields that are saying how many dots or what density of dots are there at the moment. And that affects the way in which the sampling happens. And we can imagine that the beliefs about states we might have are represented in frontal parts of the brain that deal with where I might choose to look, that the more visual parts of the brain are going to be represented at the back near the occipital cortex, or even higher order summaries of those things in parts of the temporal cortex. And the mapping from one to the other is represented by the kinds of visual pathways and white matter tracts that connect those different regions. So here we'd be looking at things like the superior longitudinal fasciculus. The equations I'm showing at the lower part here just separate out this idea that if our prior is that we're going to make decisions and make choices to sample and change the states to maximize my information gain, which is represented here as a mutual information or a KL divergence between a joint distribution and several marginal distributions, then we're both maximizing information about the states we might choose, but also about the parameters that we were talking about earlier. So with each eye movement, I make a new observation, and that adjusts this connection. It adjusts the A, or the synaptic weights that mediate my prediction of what I would see if I were to look there. You can see that in this situation, it gives fairly even sampling patterns across space, because every time I've looked somewhere, I update those beliefs and I don't need to look back there again because I've got slightly more information about it. Whereas parts of the space I haven't yet explored, there's more information yet to be learned. And neurobiologically, I might interpret that as I can update my synapses a little bit more when I make an observation over there. Now, that's a long way of getting us to this idea of pathology. So here I'm showing the same sort of same sort of sampling I was showing before, where both sides of space are sort of treated equally. They're both equally likely to update my beliefs. Let's say we now disconnect two parts of the brain, or we cause a lesion there, and we do this in one hemisphere but not the other. So typically this would be something we'd see in the right hemisphere when a stroke has happened that's affected these white matter tracts. We might interpret that disconnection as now saying that I can't update those synapses anymore. There is no connection to update. Therefore, I'm implicitly very, very confident about the connection there because there is no potential that I can update my beliefs. And so we can represent that same sort of idea by saying, let's treat the left side of space here, represented by the right hemisphere of the brain, as being incredibly confident, with the right side of space being as normal. And you see there's now this sort of very biased sampling towards the right side of space, simply because there's more information to be gained there. I'm more likely to be able to update those synapses that do still exist compared to those that have been disrupted. And this is what is seen in right hemispheric strokes that cause a condition known as visual neglect. And so we see a very close mapping between the kind of priors we've chosen in our generative model, the structure of the generative model, and both the behavior and anatomy that we might see in a pathological condition. I'll now go through another sort of example that allows us to pick out a whole series of different priors, and that's the example of motor control. And as we were talking about earlier, we can see the process of enacting motor actions as being one of fulfillment of predictions. And again, I was talking about this idea that we can see reflexes as being disruptions of those predictions that lead to actions to try and correct that. So let's think a little bit more about the structure of those predictions and the kinds of model we might use. So if we're using a Gaussian type model, a normal distribution with some function g that predicts what we're going to observe, then the log probabilities of that will be effectively a quadratic function. So looking at the squared difference of prediction errors, a difference between observed and predicted data, modulated by this pi term, which is our precision or confidence in those predictions. And that pi is going to be really important. So let's think a little bit about the structure of that. Typically, we'll have two different parts to it. One of those is a prediction about how the variance at any point in time is likely to be. So we can see that on the left, that we've got a difference between a very noisy trajectory where there's a sort of wide variance in our predictions over time versus a much more precise, less noisy trajectory. Then the other part of it is thinking about actually the temporal correlations of those trajectories, how smooth that noise process is. So you can see on the right that the amplitude is going to be very, very similar in both cases, but actually the smoothness of those fluctuations varies quite a lot, and both contribute quite considerably to the kinds of predictions we might make. So let's say we induce a lesion where we now predict that there's much less noise and much fewer fluctuations than we might expect. And I'll show you just the difference here. So once we're introducing our unexpected proprioceptive data, the tapping on the tendon, we're getting this relatively small response. But if we now assume that actually all data is relevant, we get a much larger amplitude response than we did before. And that's because it's very difficult for our brains to now say, actually, this is just noise and discount it. And so it pays much more attention and treats it as being much more salient when there's an unexpected data point. And just show you again, as before, it was relatively small. And then once we assume that the data and the noise is relatively limited, everything is interpreted as signal, and it introduces this much larger pathological response of the sort we see in people who've had disruption of the tracts or spinal cord lesions or motor cortical lesions that affect our predictions of these sorts of inputs. What happens if we look at this other sort of lesion? So here, let's overestimate the smoothness and the temporal correlations. And here we see this alternative pattern of pathology where we get a pendular oscillatory type reflex. And this is the sort of thing that's seen in lesions of the cerebellum, a part of your brain that's often associated with balance and coordination. So again, by examining the form of the generative model, we can bring out different kinds of pathology that are consistent with what happens in pathological conditions. Now, I'm just showing an example now where we take this reflex and we equip it with a much more complete model, which I won't go through in detail, but I've sort of unpacked the different bits of the message passing in terms of plausible structures in the brain that might mediate that. And by having the slightly more complex model that allows us to decide between alternative locations we might sample, we can deal with this slightly more complex coordination task where I have to move my arm to different locations, depending upon which of these three targets is highlighted. Now let's make the same lesion we were doing earlier, where we overestimate the smoothness and see what happens there. And again, we're interpreting this as being a lesion of this part of the brain, our cerebellum, as this overestimation of the autocorrelation structure in our model. And you can see here it significantly changes the coordination of our model. We end up of our behavior. We end up with these sort of over and under reaches, this sort of oscillatory ataxic type behavior that again we would see in these kinds of lesions. Let's look at other parts of the model that are dealing with more the decision making between each different alternative location. So the lesion we're making here is now between different hierarchical levels, between the fast and slow. And the key thing here is that the fast sort of adjustment when I see a new target and the ability to plan a route that reaches that target is relatively intact. But as soon as there's a change in target, as soon as this higher level, slower context changes, it takes much longer to adapt to it. The idea being that the slower order narrative and the way it communicates here with what's happening at the faster levels of inference is really important for sort of tying together sequences of episodes over time. And when we disrupt that connection, we may eventually be able to adjust, but actually there's quite a lot of difficulty in dealing with things when the context changes. And this is something that is seen to some extent with frontal lesions and this idea of perseveration, of carrying on doing the same thing, even when the context changes. And then finally, I'll show you an example. This is a lesion of something more like the basal ganglia structures I was talking about before, the deciding what I'm going to do next. And if we reduce the confidence in what I might do next, you end up with this sort of odd slowing pattern and this failure to initiate the right sorts of movement at all, of the sort we might expect to see in conditions like Parkinson's disease, with a depletion of the dopamine that projects to and influences our basal ganglia decision making, and the difference between these two different pathways I was talking about earlier, this sort of highly contextualized, slower pathway, and the much faster contextual pathway that might deal with decision-making and planning, and this sort of change of the emphasis more towards this slower contextual pathway slows down the movements that we might make. So I've shown you a few examples there, and just to recap, we sort of started from this idea of neuronal dynamics as being part of an inference process. and thought about the way in which we can sort of pin that down in even very simple sorts of physical systems and reinterpret their dynamics in terms of the effect of beliefs on behavior. That depends very sensitively upon the form of generative model we might use. And really that is the central part of this talk, of thinking about what kinds of generative models our brains might use, and therefore what sort of models might we use in artificial intelligence. And the ways in which we can measure the implications of those models in biology is both by looking at the anatomy, the behavior, and the way in which disruptions to specific parts of the anatomy might influence the behavior, and therefore what that tells us about the generative model. I'll conclude there and take questions. And I've sort of highlighted some of the people who are involved in some of the specific bits of work I've spoken about today, although there are many other people to thank in many of the other bits of work that have sort of led into this as well. I've just sort of shamelessly highlighted the Active Inference book, which is available both for purchase, but also as an open access version through the MIT Press website for people who are interested in learning more about these kinds of methods. And some of this work was sort of done at UCL and partially funded by the Rose Tree Trust, and some of it was done sort of later on in Oxford, where I'm based now, at the Nuffield Department of Clinical Neurosciences, where I've been funded by the NIHR. But at that point, I'll stop and take any questions. Thank you all for listening.
Thank you so much, Thomas. Thank you for such a great talk, really. Yeah, I would say we will first ask the audience if anyone has any particular questions. You could just type in the chat or just ask us to just unmute you, and you'll be able to ask your questions from Thomas yourself. If there are any questions.
Usually the case is people take a bit of time before they start speaking up. So maybe I could start if that's all right.
Sure.
Yeah, thanks a lot, Tom. That was great. I guess I have a couple of points, a couple of questions. One was like, it was a very nice example where you're showing graphical models where you could show PCA and CCA and even transformers as a graphical representation. I would probably add Bayesian filtering and generalized filters. They all can be shown through graphical models. However, I think there's a one key difference at least. Because, for example, generalized filtering or Bayesian filtering, they have been developed in the context of MRI, where everything is offline, or even for PCA, let's say, you have all the data that you want to look at. The Y is given, and then you look at all the inferences which is happening is while in active inference, you have to the perception, for example, is online. So I mean, what I'm getting at is how do you go from the offline to online inference, if that makes sense?
It does, and it's a really good point to make. And I suppose the key thing is thinking about which data points you have available at any point in time. And it's certainly true that some of those data points will become unavailable as they become past data, and some of those data points are future data and therefore aren't available yet. And that's exactly right. And I suppose the way you might represent that graphically is just whether or not you have that data point available for each point, and then indexing things in time. And that leads to a whole series of really interesting questions about, well, so first of all, the distinction between discrete and continuous timeframes. So you mentioned generalized filtering, which is a method that I know you've had a lot of involvement in the past, but is also a method that's very much used in a lot of active inference models, and certainly in the more continuous models that I've described today. So the arm movements, for instance, have at the lowest level of their model a generalized filtering model that uses the idea of just having a particular time point and potential or data point, but also a measure of the changes in that data point over time. So the derivatives and local information about that trajectory that are then used to draw those kinds of inferences. And in fact, that local information, so there's a bit of a diversion, but local information about not just the current data value, but also its velocity and acceleration and so on, becomes incredibly important in terms of generating those reflexive type actions, because it's being able to predict how things are changing that matters in terms of working out how my predictions can best be fulfilled by my data themselves. So that's really interesting. And the other way of looking at it is whether you're dealing with discrete time models where you're sort of at each point getting a measurement, which itself raises questions about different kinds of models that might do that. So do you, for instance, have a model that is consistent over time, but just change which data point is available? Or do you have a model where everything moves with time, where you're constantly just predicting the next thing and the previous and priors from the previous thing? And both are sort of perfectly reasonable ways of dealing with this sort of modeling. And it's interesting to think what would be the sort of different predictions and different kinds of anatomy you might see with each of those things. I don't know the answer to that. Generally, a lot of models have sort of gone somewhere between the two, where you often assume some degree of rollout in time, but also just update where the data points coming into your model from moment to moment. But yeah, no, really good question. And I think that there's sort of it's open about how you represent that. I tend to think that graphically it's easier sometimes just to represent the sequence as is. But I suppose if you were being really accurate, you might represent a whole series or a sequence of whole graphical models with each different data point becoming available in each model.
Yes, that's excellent. I mean, some of the points that you have made, they make really much sense, and it's something that we have been thinking about, and there's some work currently being done. Maybe something we'll touch base, but I see that now we have a few questions which have started to come, so I guess Fro would like to prioritize them rather than myself one another time.
We'll come back and forth maybe. Yeah, maybe I'll ask this one question from the chat from Nicholas. They're saying that they're a PhD student thinking about going more into the computational modeling and wondering what would be a good way to apply these active inference models to fMRI task data? And are there models already available, or do they need to come up with generative models from scratch?
Yeah, so I suppose the answer is both, and it depends. You know, it's very fun and flexible to be able to come up with those sorts of models from scratch, and it depends a little bit upon the kinds of tasks you have in mind. But it's also true that there are a large number of models now available and you know, sort of made open access that can be used for this sort of problem. The inversion schemes of the generative models, so the way in which they're solved, are very much available, and the process of coming up with the model itself that you then solve using these off-the-shelf schemes is much simpler than if you had to write the entire thing and all the mechanics yourself. It depends upon the tasks you're using, to be honest. So if you wanted to apply an active inference model to a particular task, you would of course want to see that that there is a model that does something very similar or can be adjusted to something similar. The places to look to see where you can find those kinds of models are there are a number of places to look now, and the sort of most developed ones are normally through the SBM package in MATLAB, which may be quite useful for somebody who's already doing functional imaging research. And just practically, if you go into the if it's on your MATLAB path and you type in DEM in capital letters, you'll get a graphical user interface that will walk you through the models that are available there. most papers that are presenting active inference models will also direct you to the specific repositories where their model has been designed and made available. A few of the examples I've spoken about are in a slightly separate repository with a more neurological focus. There's also a Python implementation of these kinds of routines, which has a few examples built in called PyMDP, which has largely been put together by Connor Hines and some of his colleagues. That's also freely available and has the advantage that you don't need a MATLAB license to use them. And I know that that there are people at Bert de Vries group in the Netherlands who've put together a whole series of Julia-based implementations using factor graphs quite explicitly as a way of doing active inference. So there's a lot available there. There's also really interesting questions about how you apply it to the MRI data themselves, because the active inference models will give you the behavior and the belief updating. And the behavior is really useful if you want to fit that to the behavior of your participants in the task, so that you can work out which sort of priors were they using. And from that, you can work out what is the sort of Bayes optimal belief updating that you'd expect to have occurred during that trial. If you've got that, then you can use that trajectory either as a sort of computational fMRI type regressor. So you can actually use the belief updating itself as a regressor to see which bits of the brain may be doing that sort of updating. Or you can make predictions about the relationships between different areas of the brain that might then be amenable to either functional connectivity or DCM type analyses, and sort of use the conceptual element of that to make predictions there. So, for instance, the visual neglect example I gave you today sort of makes predictions about the way in which connections between the frontal and temporal parts of the brain might update, even in health, when sampling different parts of visual space. And that's a prediction that we then took forward to an MEG study, which I haven't spoken about today, where we got people to do exactly that paradigm and found evidence for modulation of those fronto-temporoparietal connections using DCM, even for each saccadic eye movement and each fixation they made. So we can actually measure the changes in that connectivity over time. So I hope that's helpful. There are a number of different ways you can use it, but certainly there are models available. But there's also now quite a lot that you can use those models as templates to develop your own kind of paradigm and make it match what you've asked participants to do in fMRI yourself.
Yeah, thanks for that. They're saying it was very helpful. I guess we have another question almost in the same space from Adam. He's asking, what's the approach to deciding the structure of the generative model, particularly defining the goal function and the hidden causes? And what are the methods for comparing the different possible models?
So the sort of question of the design of a generative model is a big question, and in a way it is the key question of neuroscience from my perspective, and I think probably from an AI perspective as well. So the approach to doing it, there are some generic principles that you can appeal to. So one of those is thinking about what is the kind of data you're trying to you think the brain is trying to explain, and are those data categorical data or are they continuous data? And that tells you quite a lot already about the sort of model you need to deal with, whether you're dealing with categorical Dirichlet type distributions or whether you're dealing with something more like the generalized filtering that Adil mentioned earlier. And from that, you then sort of ask, what sort of behavior are you trying to explain? So are you trying to describe a movement trajectory, again, much more continuous, or are you trying to look at a more complex planning type problem from the way the question's formulated in terms of the cost functions and goals? I think probably we're talking more about a planning type problem. And so there's a fairly generic way of describing the kinds of goals you would deal with in active inference in terms of an information theoretic quantity known as expected free energy, or sometimes a generalized free energy. and it's expected or generalized in the sense that it deals with future data points that are yet to be observed, whereas a sort of more classical variational free energy is, or an elbow from a sort of machine learning perspective, is just a way of describing marginal likelihoods of data you already have. That's important as well, in that marginal likelihood answers the second question of how do you compare the different models. Now, the expected free energy is made up of well, it's one thing, but it can be decomposed in different ways to highlight different perspectives. And one of those is to separate it out in terms of cost functions of the data themselves. And that's the kind of thing that's more familiar to people in reinforcement learning, Bellman optimality type principles, value functions, expected returns, and all that sort of thing. The other part of it is the more intrinsic motivation that I was speaking about earlier, the idea of curiosity. It has some links to ideas like empowerment that people like Daniel Polanyi have put a lot of work into, where it's actually the belief states themselves that have an impact in terms of what you do, and you're almost trying to optimize the belief states rather than just optimize the data. The particular way in which those work will itself depend upon the generative model you have in mind, what things there are to be uncertain about, what sort of uncertainty and opportunities for uncertainty resolution exist in your model, the space of alternative behavioral choices you can make, the depth of the plans and policies that you have in mind. And I guess the ultimate point about the cost functions is what sort of data do you expect to observe? Now, cost functions are a really interesting concept, and stop me if I'm going on too long with this answer, but it's quite a big question.
Yeah, please go ahead.
The cost functions are really interesting in themselves in terms of and reward functions, in terms of thinking about what they actually mean. And really what they're describing is the behavior that leads to them. So there's a real circularity in definition, that the reason something is rewarding is because you expect to go towards it. You expect it to be attractive and to behave such that you get it. And so saying the behavior is for the purpose of maximizing reward is itself a very circular idea. And that's quite a useful perspective on it, because you can start to reinterpret the question of what is rewarding in terms of what sort of data do you expect the kind of system you're developing to experience most of the time, or to act to get towards if it deviates from that. So if you're designing a human, you might expect them to be satiated and not hungry, for instance. And so just on the assumption that you're not hungry most of the time, you might generate a model where being full is more likely, and then any behavior that deviates and leads to being hungry will be met by a response that like cooking yourself lunch and having something to eat. So it's an almost circular way of defining it. In terms of how you then, if there's ambiguity in that and there's not an obvious way of defining it, then ultimately it comes down to a question of treating your different cost functions as being different hypotheses, and potentially other bits of your model as being different hypotheses. And if you can write down your model, simulate the data, fit it to empirically observed data, then just as you would with a dynamic causal model or any other statistical model, you can then compare those using a marginal likelihood or a free energy or an elbow to then do a formal model comparison and test which of your hypotheses best explains the data you have at hand. And that's the fun of science, really, isn't it? That actually, if everything were really obvious how you'd write it down to begin with, it wouldn't be science at all. It's only when there's some alternative ways you could do it, and it might make sense, that you can then compare it with those data and do those model comparisons.
Thanks a lot, Thomas. I guess I don't see any other questions from the audience. I have two questions myself,
Yeah.
but for the sake of time, do you have a question, Mike?
Yeah, but go ahead. Yeah, maybe in virtue of time, I have also a couple of questions, but maybe only ask one question.
One each, yeah.
Yeah, one
Yeah.
each.
Okay.
Yeah, maybe I'll go ahead. You may have already basically touched upon this, and I'm just asking this to make sure it's clear for myself. But I was wondering how does basically the framework active inference can distinguish between these. You had these wide range of examples, right? Some of them are disorders that come from impaired priors, like the visual neglect, versus those that come from let's say misspecified likelihood mappings, like the noise, the sensory noise that you get in Parkinson, for example.
And I suppose so the question of whether it's misspecified. So in effect, actually, you could argue that the structure of the model and the particular parameters of the likelihood you're assuming are themselves priors. That I'm putting a prior on the parameters of a likelihood distribution.
Yeah.
So in a sense, it's always due to a disorder of a prior. Now, I suppose, sensory noise itself is not necessarily different in Parkinson's disease, but your estimation of that noise might be, which is a sort of key distinction. So people often talk about sensory noise being greater or lesser. That may be true in some sort of peripheral neuropathies or disorders of the sensory apparatus themselves. But actually, most of the time, what they mean is that the way in which I'm estimating and dealing with that noise is not right. And so that comes down to what priors I put on that, and those may be priors of a sort of overall variance. I often think about these as almost like a spatial and temporal component. They may be the spatial sort of moment-to-moment variance, or it may be the correlations over time, the autocorrelation functions, both of which then contribute to that. And the priors you put on those two things has a very profound effect on how you end up generating movements.
Yeah. Yeah, thank you. Thank you very much.
But ultimately, in distinguishing which one of those might affect it, you can either do a forward simulation and say, if I vary this parameter, what's the variation in behavior I would expect? Or you go back to the data again and you say, well, can I fit this to some data I have, if you're lucky enough to have the right data features for that, or the right kinds of data in the right patient populations. And you can then start to ask, well, what values of these parameters are the best explanations for the behavior I'm seeing?
Yeah, makes sense. Thank you. Thank you very much.
Great. Yeah, maybe I can ask quickly. So in the visual sampling slide, there was an equation that shows the decomposition of expected free energy into information gain about states versus parameters. So yeah, if we can.
It's going a bit slowly, but I'll get there. I know which one you're talking about.
If you,
There we are.
this one. We have two terms, information gain about states and about parameters. So do you think both components are simultaneously optimizing visual behavior, or do they operate on different time scales? For example, perceptual inference versus learning.
Yeah. Well, I suppose these are both influencing process of planning rather than directly the process of updating. So there are probably different timescales relevant for the updating of these terms. That you might expect that it's easier to update the states or your beliefs about the states quite quickly once you've made a new observation, whereas it might be that the parameters end up being updated more slowly. In this particular model, actually, they are updated at the same timescales. And in fact, that seems fairly consistent with what we ended up measuring empirically, that actually for each fixation, there seemed to be an increase in the connectivity strengths or the effective connectivity. In fact, we found a decrease in the inhibitory connections in the other direction. So you end up with a sort of facilitatory effect overall. So yes, you're right. I think there probably are different timescales of perceptual inference and learning. Probably both have a pretty similar effect on the process of planning, because you want to get data that are relevant for both. Now, of course, if you are really uncertain about the states, then knowing how much information you're going to gain about parameters is itself quite difficult. So that also has a key impact on how these two different terms update. So you can see that the second information gain term is an average or expectation under posterior beliefs about the states of the world. So that's very uncertain. If I don't know which states I'm even going to encounter, then that information gain term for parameters might be the same for all the different policies I could adopt, in which case it has no relevance at all. Whereas the one about the states will have much more relevance. And I guess that's probably the key in terms of how you're thinking about this, that actually once you've sampled information that lets you pin down what the states are, then you're in a better position to take action that will resolve your uncertainty about the parameters. Of course, you could make exactly the opposite argument and say that it's almost impossible to know how you're going to gain information about states until you know what the parameters are. So, you know, it gets tricky there. But I think you're probably right that over time, if there is a clear distinction between these things, probably the information gained about states will dominate earlier on. And then once you're more confident about the states, you're probably in a better position to resolve information and uncertainty about the parameters.
Great. Thanks.
Yeah, thank you very much again. If I mean, probably everyone has more questions, but for the sake of time, we're already past an hour. So if there aren't any burning questions from the audience, once again, thanks a lot, Thomas. Definitely, we can be more in touch with our other questions. I will be very interested to know your perspective on whether do you think these can be applied in our embodied in vitro neuronal cultures, kind of finding equivalence to the emergent phenomena that we see here in those networks. But maybe we can chat offline more.
Sounds good.
Yeah. Thank you very much.
Thank you very much.
You bring a lot of excitement to these. Thanks a lot. Yeah. Great.
Thank you so much.
Thank you very much, Thomas, and thanks everyone for joining us today.
Yeah.
Bye.
Have a great day.
Bye.
Bye then.
Bye.
Bye.